Hi again. So today we're going to continue with the microminerals and move on to copper. So copper is found in really small amounts in the body, just 50 to 150 milligrams. So it's kind of distributed everywhere in the body like zinc, but pretty small amounts. And when I saw that number, I kind of wondered how it compared to a penny. So a penny is 3.1 grams, which is basically, what would that be? 3,000 roughly milligrams. So basically it's just a fraction of what the weight of a penny is. So we have nowhere near kind of a penny's worth of copper in our body. But yeah, just a fraction of that. So really, really small amounts kind of distributed everywhere. And copper in the body is found in two valent states. So we'll notice in the absorption slides that it's found in both the 1 plus state and then the 2 plus state. And usually higher valence states, kind of what we saw with iron too, are found outside of cells. So in the 2 plus form, that's kind of how it comes in foods and stuff. And then when it's in cells, it's in the 1 plus state. So you'll see that when we get to the absorption slides. And that was the same with iron. Remember, it was in its higher states, kind of it could be in the 3 plus state outside and then 2 plus state inside. And then 3 plus again when it was traveling to different tissues of the body. All right, so food sources of copper are pretty similar to that of zinc. So you could also say like meats and seafoods. That's kind of the major sources. The RDA for iron is 900 micrograms for men and then 900 micrograms for women. So it's the same. And then if you kind of go down the list here, you'll notice that usually like a serving of kind of like seafood, oysters, you pretty much get, definitely not as high as zinc, but you get pretty much all your needs for the day. Liver, again, provides super high amounts of pretty much everything, just because that's one of the main places we store a lot of vitamins and minerals. So any kind of 3-ounce serving of liver is going to give you kind of a pretty potent, kind of high amount of that nutrient. But we also find kind of moderate intakes in like beans, nuts, and there's even a little bit in chocolate. So 1 ounce of, this is more like dark chocolate, that's about like half a bar usually. I guess it depends how big of a bar you're getting. But like, I looked it up, like a Hershey's bar, just to give you a sense, is like 1.5 ounces. So if you're doing more of the kind of nicer dark chocolate bars, those are usually like 2 ounces or so. So if you eat that whole bar, you're getting actually 400 micrograms of copper. So yeah, quite a bit of copper in chocolate. Do you remember what the other nutrient in chocolate was? It was magnesium. So dark chocolate's a really good source of copper and magnesium. The amount of copper that you're going to find really depends again on soil. So very similar to what we talked about for zinc. We're overusing the land. We're not giving soil time to kind of recuperate. So we're basically planting crops on it every year when we should be waiting a couple of years between plantings, I guess, but we don't do that. So basically, a lot of our soil has pretty low levels of copper and zinc. So not like zinc is way lower. Copper it's okay, but it could definitely be better. The other thing is preparation and processing. So if you remove the husk of the grain, so that outer portion, you're gonna kind of lose a lot of the copper, kind of like with a lot of the other minerals. So you definitely, always the whole grain versions of whatever are definitely have a lot more nutrients in them. Copper does have something, like with cooking in general, it kind of acts like a water-soluble vitamin. So if you're like boiling whatever beans or something, you can actually lose about 50% of the copper because it will seep into the water because it is kind of water soluble. So yeah, kind of seeps out there, and then the same thing, you want to recoup that boiled water and try to use it in whatever you're making, like soup or I don't know, whatever it is. But yeah, to kind of recoup some of that copper. Food processing, I mentioned it in the previous slide, but more specifically, since most of the copper is found in that husk, one of those outer layers, When you take off and refine grains, you can lose up to about 70% of copper with refinement. So again, yeah, I'm sure you all know as nutrition students, but yeah, white bread or the refined breads are really not great. Copper supplements, copper's not really something that's like heavily, there's not really that many deficiencies in copper actually. Our bodies are really good at recycling it. So it's almost hard to find copper supplements when you go to like Target or Walgreens or wherever you go to look at supplements. It's just not a really common thing. Even before taking this lecture, it's like it's hard to even think of like what does copper even do in the body? Like people don't really supplement with it for like colds or lowering heart disease risk or anything like that. Its functions are actually pretty hard to remember. They're kind of these nitty gritty kind of enzymatic functions. But yeah, so I always struggle to find any supplement bottles at all. But just looking in, I kind of did a bit of research. If somebody does need to supplement with it, copper carbonate tends to have the highest amount by weight. And there's also been there's some data to show that it's also the best absorbed. So yeah, I'd probably recommend copper carbonate above the other forms. The supplements usually have about 2 milligrams in them, so that's 2,000 micrograms. And remember, the RDA for men and women is 900 micrograms. So each of these pills is going to give you about twice the RDA. But most of the time when we get to kind of the deficiency slides, a lot of people that have copper deficiencies tend to have problems just absorbing copper in the intestine. So if somebody, realistically, if you run into somebody with a copper deficiency problem, you're probably just going to have to do intravenous injections because the reason that they have the deficiency is because of absorption problems. So yeah, just keep that in mind. And I mentioned before the reason, even though our soils are mildly depleted in copper, the reason that most of us don't have deficiencies is because our bodies are really, really good at recycling it. Copper is found in pretty much every cell of the body, kind of like zinc. And then we kind of like reabsorb and reuse it. But we kind of secrete it in our saliva, gastric juice, pancreatic juice, duodenal juices. So yeah, it's kind of just like constantly released and then it gets reabsorbed. In foods, copper is bound to proteins, so amino acids. When it's kind of liberated from the protein source, it turns into the 2+ state. And then in the small intestine and in the stomach, basically the way that, kind of same as zinc again, we need to kind of cut it off of those proteins. So release it from the amino acids. And that's done by HCl, pepsin, and proteases. And yeah, that's done all throughout the stomach and the small intestine. And then, yeah, what we end up with again is the 2+ state of copper. So try to kind of follow when the copper changes from valence state to valence state. Copper is absorbed by 2 main processes. Again, most of the minerals, kind of, I guess we've done a lot of them at this point. You'll notice that for the most part, they do follow the water-soluble vitamin kind of typical thing. Active energy-dependent transport with low to moderate intakes, and then passive diffusion at higher intakes. So just a bit more information. First of all, the energy-dependent are the active carrier-mediated transport systems. So copper is absorbed into the enterocyte by 1 of 2 receptors. So there's 2 receptors present on the brush border, kind of ready to take in copper. The first one is called HCTR, and it's I think it's just like copper transporter, but I couldn't figure out what the H stands for. Couldn't find that. Everybody just refers to it in all the textbooks as HCTR, but yeah, probably copper transporter. And then also DMT1. So both of these things are taking in copper in the 2+ state, remember. So where did we see this one before? Divalent mineral transporter 1, DMT1. What else is transported like that with that receptor? It is iron. So iron and copper compete for this, but iron is generally better absorbed through that. So yeah, just keep that in mind. But yeah, copper can basically enter via both these receptors. Then once it enters the enterocyte, it's pretty much immediately reduced to the 1+ state, so both times. And this is done by copper reductase, which is basically sitting kind of right adjacent to these receptors. So it kind of immediately gets reduced down to the 1+ state. And then within the enterocyte, it's chaperoned around by 3 different transporters. And these transporters, depending on what it binds to, those transporters direct copper for specific uses in the cell. So if it ends up binding to the glutathione, it tends to just take the route where it just leaves and gets distributed to the rest of the body. If it binds to ATOX1, that means that that copper is going to be used to make superoxide dismutase. So that's one of the functions of copper. And then if the copper binds this third transporter, COX-17, it means that the copper will then be used to synthesize cytochrome C oxidase. So yeah, depending on what it binds to, that kind of directs what it's going to be used for. Copper is transported then out of the enterocyte. So remember, when it binds to glutathione, that means that it's going to be distributed to the rest of the body. And then so the receptor, or the kind of transport protein, or however you want to say it, that helps it leave the enterocyte on the basolateral side is ATP7A. And then this plays an important role when we get to the kind of deficiency toxicity slide. So just remember, ATP7A is the one that helps it get out of the enterocyte. And then the second way that it's absorbed is just, at higher intakes, passive diffusion. So yeah, just absorb passively, and then it basically follows the same route. That reductase is there to reduce it down to the 1+ state, and then it does have the option of binding to glutathione, ATOX1, or the COX-17. And then percent copper absorption on average ranges it's about 50% that's absorbed, but then of course, it totally changes depending on intake. So if you take in really high amounts, so maybe a couple of those supplements all at once, up to like 5,000 micrograms per day, the absorption will decrease to 20%. Whereas if you're having kind of low intakes, or just kind of moderate intakes, whatever you'd find by consuming, like nuts or beans or whatever it was, or even some of the seafood sources, absorption would increase then. So the 50% is just an average. But, this follows the same rule. I hope, this is one of the main things that I wanted you to learn in this class, is that, the more you bombard your system or your gut with nutrients, the less it's going to take in. You basically just use up all those receptors. There's not enough receptors to bring it all in. They get overloaded. So most of the stuff will be excreted. So if you want to kind of replenish someone's system or get most of the nutrients in, the best thing to do is take small amounts in more frequently, like every 3 or 4 hours or so. So absorption inhibitors, high intakes of iron. So I'll explain that a bit more in a second. We kind of talked about it briefly. High intakes of zinc. Remember, toxicity of zinc. What was the toxicity effect? Copper deficiency. And I'll show you the mechanism for that in the next couple slides. And then another absorption inhibitor for copper is vitamin C. So remember, when you liberate copper from the protein sources and foods, it's in the 2+ state in the gut lumen. And that's the state you want it to be in, so it can then, get absorbed by those various mechanisms. But if it happens to interact with vitamin C in the gut lumen, vitamin C will reduce the copper to the 1+ state, and then the copper can't be absorbed because those receptors that I showed you, so these guys up here, need it to be in this 2+ state for it to basically go in here. So if it's in the 1+ state, basically, if vitamin C out here interacts with this, turns it into the 1 plus state, then just can't get in, so will just be excreted. All right, so absorption enhancers. So if you have a diet with kind of higher protein, so amino acids, and then acetic acid. So where do you find acetic acid in foods? Vinegar. So that's kind of a component of vinegar. So both those things, kind of higher protein, or if you have like vinegar in your foods, that increases solubility. So that just means that you can basically break copper off of those protein sources easier. So yeah, more is absorbed. So just to go into a bit more detail, I know we talked about it a bunch of times or mentioned it a bunch of times. So how does kind of high intakes of zinc, so if you're maybe taking zinc for cold, as a cold remedy or something like that, how do those high intakes of zinc decrease copper status? So the main thing that happens is a high intake of zinc, if you're taking that in, that stimulates your body to create more metallothionein in the enterocyte. And so remember, metallothionein is the storage form of zinc. So you're taking in lots of zinc, the enterocyte senses that, starts making a lot more metallothionein to store the zinc. The problem with this is that for some reason, the enterocyte metallothionein, so this is specific just to enterocyte metallothionein. I'll show you in a couple slides. In the liver, it's a different picture. But enterocyte metallothionein binds copper more avidly than zinc. So if you're taking in copper at the same time, like, they have shared food sources like meats and seafoods, what will happen is that the copper will basically get trapped. It binds more avidly. It immediately goes into the metallothionein. And then the problem is it can't get out. So it gets in there. The body's making metallothionein to store zinc, but then by accident it ends up trapping copper in there. And then once it's in the enterocyte metallothionein, copper cannot get out. So basically it's just excreted when those enterocytes are kind of just sloughed off naturally. That's generally how high intakes of zinc can lead to a deficiency of copper. And then another interaction that we kind of briefly mentioned is iron and copper. So remember, iron can inhibit copper absorption, and that's because both of them use that divalent mineral transporter 1. The problem is iron has a much higher affinity for it, and so if you're taking in copper at the same time as iron, basically iron will kind of push its way forward and then just use up all the DMT1s, and copper will just kind of be stranded and have to, it won't really have a chance to get absorbed, so it ends up getting excreted. So that's how high amounts, if you're taking in iron with copper, it's usually like supplemental levels of iron can lead to copper deficiency in that way. So after leaving the enterocyte, copper is bound to albumin. It travels through the portal circulation to the liver. And then this slide always confuses people a little bit. So once it enters the liver, it binds metallothionein. So liver metallothionein is different from the enterocyte one. For some reason, they're not quite sure. Again, I don't know how much research has been done in this area, but metallothionein in the liver is somehow able to let go of copper, whereas the enterocyte one is not. But basically, it's kind of this like middleman where copper comes in, attaches to metallothionein. It's then transferred to something called apoceruloplasmin. That's kind of like a precursor to ceruloplasmin. And then from the apo, it's transferred to ceruloplasmin. And then this is kind of the way that we transport copper to the rest of the tissues. So from the gut, it's transferred from the gut to the liver by albumin. And then the transporter for copper from the liver to all the tissues of the body is ceruloplasmin. And ceruloplasmin has actual, other than just transportation, it has some unique functions as well. So very small amounts of copper are found all over the body again, just like zinc, super ubiquitous. It's kind of found everywhere. So main functions are really kind of like enzymatic functions. It's basically a part or involved in ceruloplasmin. We'll talk about those functions in a second. It's also in SOD, superoxide dismutase. So where did we see that one before? That we saw that in the zinc lecture. Cytochrome C oxidase, so it's in the electron transport chain. Dopamine monooxygenase, and then it also has a role, a non-enzyme role in gene expression. So just to tell you, so ceruloplasmin basically helps transport copper around, but it also has its own kind of proper functions. One of the main things that it helps out with is oxidation. It has two functions. So the first one is oxidation of minerals. So it basically helps, if you remember, where did we see this before? If you remember, we saw it in the iron slides. So ceruloplasmin helps convert the 2+ iron to 3+ iron so it can bind transferrin. So we saw, if you go back to your iron slides, you'll see that we mentioned ceruloplasmin when we're looking at the basolateral side. So basically when iron's bound to ferroportin, that receptor that kind of helps it get out of the enterocyte, and then right before it binds to transferrin, that's the transport protein for it. So ceruloplasmin is the thing that converts it from the 2+ state to the 3+ state to basically help it bind to transferrin. Because remember, with pretty much all these things, they're in the lower, if they do change valence states, they're in the lower valence state inside cells, and then the higher valence state outside cells. So iron is in 2+ in the enterocyte, 3+ outside of the enterocyte, traveling to other tissues. Copper is in the 1+ in cells in the enterocyte, and then the 2+ when it goes to, travels to other tissues. Ceruloplasmin is also involved kind of like in an immunity type role. It's involved in superoxide radical scavenging. So when someone has an infection, remember the main thing that happens is usually macrophages perform this like phagocytosis of invading organisms. And then it just so happens that when that's happening, that whole phagocytic process generates superoxide radicals. And it just, one thing I didn't realize until putting these lecture slides together is ceruloplasmin is kind of there. It kind of gets, one of the other things that kind of gets recruited during these immune responses. And it kind of just hangs out there because it helps to eliminate these radicals. So yeah, the exact mechanism's not clear, but it's just recruited and basically helps clean up kind of the what's going on in the immune process, clean up any superoxide radicals that are made. So another totally separate function of copper is its involvement in superoxide dismutase. So remember that is involved in, or basically it's found in the cytoplasm of cells and then also circulating around in the plasma. So it's found, copper, remember, is, well, at this point it's found in the 2+ state. And copper is actually found at the active site of this enzyme of SOD, so where the substrate binds to it. And then removal of copper can actually, so if you have a copper deficiency, one thing that you might also see is reduced SOD activity. So kind of just keep that in mind too. So what is SOD doing again? Just remember, so basically copper and zinc, because this one is also a zinc-dependent enzyme, basically are components of this for the most part. And what they do is they basically help get rid of superoxides. So, which are these guys? So SOD is present, remember, inside the cell, then also floating around in the plasma. And you want to get rid of these superoxides because if you didn't, they would go on to damage components of cell membranes. So there's the superoxide there. And then what would happen is they eventually form hydroxy radicals. And then those hydroxy radicals can damage either DNA or lipids. So you definitely want to kind of shut that down. And then the way that, sorry, just going back a couple slides, the way that superoxide gets rid of that is basically by making it into these two inert things. So just oxygen and then hydrogen peroxide, which then later become water. So yeah, kind of just tries to get rid of them before they can cause any damage to cell membranes or DNA. Yet another function of copper is cytochrome oxidase. So it's involved in the electron transport chain. Specifically, it's the last enzyme, so cytochrome C here, the last one in the electron transport chain in mitochondria. And then this enzyme transfers an electron. So copper assists with this to basically result in the reduction of molecular oxygen to form water. And then that generates ATP, or enough free energy to permit ATP production. So yeah, just know it's in the electron transport chain as well. Copper is also a component of dopamine monooxygenase. So remember, what is dopamine? It's basically a neurotransmitter. So, and remember, dopamine monooxygenase is an enzyme that converts dopamine to norepinephrine. And so if we look first at dopamine, remember that's the one that plays a major role in like the reward kind of motivation behavior. It's kind of like people call it like a feel-good neurotransmitter. And then also remember, neurotransmitters are just those things that send messages between nerve cells. So that's, all these things, that's what they're doing on kind of a basic level. And then norepinephrine, on the other hand, we've already talked about this, I think, a couple times in this class. But remember, that's that stress hormone involved in fight or flight. It acts on the brain. It's the one that kind of like gets your heart pumping. It, increases blood flow, etc. And then it also helps release glucose from energy stores. And then it's made from dopamine. So, does anyone remember this slide? This slide is from basically one of our very first lectures. So it's from the vitamin C lectures. So here, remember we talked about copper in the vitamin C lecture when we were talking about hydroxylation reactions. So dopamine to norepinephrine, remember hydroxylation reaction, adding this hydroxyl group. Enzyme for that's dopamine monooxygenase. Cofactor is copper. And then what recycles the copper? Vitamin C. Yeah. So basically, kind of just remember back, we did talk about one function of copper a long time ago. And then the last function that I want to discuss with you is in gene expression. This is a non-enzymatic function. Basically, copper will bind to specific transcription factors, and then those transcription factors then bind to promoter regions of DNA, and then, that basically enhances or suppresses transcription. But just know that it itself it's also involved in gene expression. So excretion of copper is, interestingly, it's pretty much entirely the bile and feces. So with a lot of these, we kind of saw that it was primarily urine for the most part, or maybe it was like skin cells or menstruation, or there was a whole bunch of different things. But with copper, it's pretty much bile and feces. That's the way it's excreted. And excretion is dependent on dietary intake. So if you take in, obviously, smaller amounts of copper, you're going to excrete less. Higher amounts, you're going to excrete more. And then just remember, our bodies are really good at recycling copper. That's why a lot of us don't have deficiencies. Even if our soils are kind of depleted, our body will just basically kind of reabsorb it in the gut lumen. But, we can lose small amounts in like urine, sweat, and menstruation and stuff. But 95% of it, most of it, is excreted in the bile and feces. So RDA is, again, 900 micrograms per day for both men and women. Kind of one of the shocking things here is that we actually didn't really realize copper was even necessary until the 80s. So again, just kind of showing you like how new nutrition research is. So basically like 40 years ago, we were like, oh, I guess our bodies also need copper to do those like 5 functions. And this was all based on, generally it's always like a balance between how much copper, like dietary intake, how much is absorbed and how much is lost. But in general, that's how much we need, 900 micrograms per day, and it is an RDA. So copper, as I mentioned before, our soils are kind of depleted, but copper deficiency just kind of if you look state to state, is pretty rare. So here, the most deficient states are in gray, but that means that less than, percent of population that consumes the RDA for copper by state. So less than 83% in the gray states. Illinois here, about 85% of us are consuming the 900 micrograms per day. So for the most part, we're fine. And then kind of like the coastal states are consuming the most amounts. And people that are at risk for deficiency are kind of the typical, elderly because of low copper intake, decreased absorption. One of the main people that are at risk for it, though, are people that take in tons of these zinc supplements for cold treatments. So they have, remember that whole taking in too much zinc will lead to copper deficiency. And now it's because the copper gets trapped in metallothionein in the enterocyte and then excreted. People taking antacids are another group that increases stomach pH, and then you have less HCl activity. So basically, you can't break copper off of those proteins. That's what you need the HCl for. So you absorb less copper. We already talked about elderly, and then malabsorption disorders as well. So people with like IBD or any type of malabsorption disorder, but particularly Crohn's, ulcerative colitis, they can definitely develop deficiencies. So symptoms of copper deficiency, you have this really interesting kind of couple clinical symptoms that are pretty apparent. I'll show you depigmentation of skin and hair. I'll show you on the next slide exactly what that looks like, but people kind of develop this kind of pale hair. And then impaired immune function, because remember, ceruloplasmin is involved with that phagocytosis process, kind of cleaning that up. But that's one thing that they've noticed in copper deficiency. Might also be related to gene expression. And then iron deficiency anemia. Why would copper result in iron deficiency anemia? So copper and iron have kind of a complicated relationship. Remember, iron can kind of lower copper intake because they're both competing for DMT1. But then copper kind of gets back at iron in the sense that, remember, why is copper needed? Where is it needed for iron absorption? So remember, ceruloplasmin helps iron get out of the enterocyte. So ceruloplasmin, remember, makes that conversion from the 2+ state to the 3+ state of iron. So iron can then bind to transferrin. So if you have low amounts of copper, it kind of like, iron can like reduce the amount of copper coming in, but then it kind of like blows up in the face of iron in the sense that now iron can't get absorbed either because it can't get out of the enterocyte because iron needs copper in the form of ceruloplasmin to help convert iron from the 2+ state to the 3+ state so it can then get out of the enterocyte and bind transferrin. So that's how iron deficiency anemia can occur when you also have with copper deficiency. So deficiency, this is what it's called. It's actually called Menkes disease. Mainly you're going to see it. If you have kind of like full-blown, you're probably not going to see these clinical features unless people have genetic disorders that lead to copper deficiency. From what I've seen, I haven't seen this kind of level reported or these clinical features reported with just people that kind of have low levels of dietary intake. So the deficiency disorder, primarily genetic disorder, is called Menkes disease. And basically what happens clinically that you see is you get this kind of kinky, colorless hair. And then in kids too, primarily affects kids because they're born with this, obviously. It also leads to developmental disabilities. And then the reason that basically what the genetic disorder is, what's leading to the copper deficiency, is there's a mutation in the ATP7A gene. So where did we see that? ATP7A. So that was the transporter that helps copper. So this one helps copper get out of the enterocyte. So yeah, the one that basically helps it get out. And then so if copper can't get out of the enterocyte, it eventually just gets excreted into the gut lumen, sloughed off cells. But yeah, so copper can't get delivered to tissues if you're not properly making ATP7A. So yeah, Menkes disease is an inability to make that transporter to get copper out of the enterocyte. And then, yeah, the clinical features is kind of this like kinky, colorless hair. It's associated with hair loss and kind of this mild dermatitis as well. It's kind of noticed very early on in children. And then the way that you treat it is daily injections of copper. So it's, when you have a deficiency disease because you can't absorb it properly, then obviously you'd have to bypass the GI tract and then just go right to intravenous injections to treat the deficiency. Toxicity is 10 milligrams per day. So that's about 10,000 micrograms, so about 10 times what the RDA basically. Acute symptoms, so people that take in, like, I guess this is 64 milligrams, I guess this is a case study in a hospital, basically involved, the main thing that happened was nausea, vomiting, and liver damage. The liver became overloaded and bloody diarrhea. But chronic symptoms also, so people taking in kind of higher amounts, 40 milligrams per day for 2 years, resulted in liver failure. So in general, I actually have never met anyone that's taken copper. I don't know if you know anybody that is really into supplementing with copper, but if you do, please email me. I'd be interested to see. But yeah, copper is one of those things that we kind of forget about. We kind of even forget it's one of the nutrients that we need. But yeah, I guess some people are really into it and they like to supplement with it daily. But don't do that because it can lead to liver failure. So the thing that I usually remember the most about copper is this interesting toxicity disease. And so this disease is also a genetic disorder, so it doesn't happen with just taking in high amounts of copper regularly. It's called Wilson's disease. And basically it results in problems with excreting copper into bile. So we can't get rid of the copper well, and then copper basically accumulates in the liver, which then of course can lead to like liver failure and liver disease. But one of the main things that you'll see with this disease is you actually see copper accumulating on the outside of the eye, on the outside of the iris. These are called Kaiser-Fleischer rings. And yeah, so if somebody has this disease, it's like very, very obvious. And one of my previous students that I taught in this class went on to do her dietetic internships, and this is one of the first people she was actually interacting with, and she's like, oh yeah, it really looks like that. So, you might actually see these people in your clinical practice. So again, it's a genetic disorder of copper toxicity. It's called Wilson's disease. So yeah, you don't get these rings again if you just take in super high amounts of copper. But you will get liver disease no matter what with both of these if it's left untreated. And then the way that they treat Wilson's disease is avoiding high copper foods, so seafoods and meats. And then to actually help get rid of the copper, they administer zinc supplements. So they're kind of like making, somebody get like a copper deficiency from higher amounts of zinc. All right, so let's move on to the mini review. So what are food sources of iron? So first of all, heme iron. Where do we get that? Animal products. And then non-heme iron. So plant products. Some functions of iron. What are some of the ones that you remember? It's involved in hemoglobin, myoglobin, oxygenases, and cytochromes. And then deficiency, iron deficiency anemia. What are the 3 stages called? Do you remember? So it's basically iron depletion, iron deficiency, erythropoiesis, And then only the last stage is called iron deficiency anemia. Toxicity, the genetic disorder is hemochromatosis, but remember, iron's probably the most toxic mineral. Kind of high amounts will get kind of everywhere and lead to organ failure. All right, so food sources of zinc. It's meats and seafood. And it's kind of the same for both copper and zinc. Well, I didn't put copper here, but I will in a second. Functions of zinc. So remember, there's lots and lots of functions for this one. I think there was like 9 or something. But ones that I always remember are taste. It's involved in immunity. It's involved in kind of gene expression. Remember the zinc fingers, the jazz handsy kinds of things. Yeah, bunch of, it's involved in SOD. So a whole bunch of different things. And then, and then deficiency. What's the deficiency of zinc? So if you don't have it, those are the ones that are still related to the function. So it's kind of immune problems, taste perception. The main thing that you're going to see clinically, though, is very similar dermatitis, like what you would see with the B vitamin deficiencies. So kind of like around the mouth and kind of like scaly skin. And then what is toxicity? We talked about this a lot today. Taking in too much zinc leads to copper deficiency. All right, so last one, copper, food sources. It's meats and seafoods. Function of copper, again, a lot of things. It's involved again in SOD. Gene expression is another one. Ceruloplasmin, helping out with iron absorption. Deficiency, what's that one called? That's the kinky colorless hair one. So it's a genetic disorder. It's the primary deficiency state. Do you remember? It's called Menkes disease. And then what's the toxicity genetic disorder of copper? That's the one with the eye rings, basically. So that one's called Wilson's disease. All right. And that's actually all I have for today. So have a great rest of your day, and I'll be posting the next lecture soon.